Okay, we will start. Next speaker is Dr. Valasek. He is from Czech Academy of Sciences in Prague. He is a faculty at Institute of Microbiology. Before that, he received his PhD from University of Vienna, and he did his postdoc under Alan Ennebusch at NIH. His lab is mainly interested in different aspects of gene regulation at translation level, which he's going to explain.
Actually, I'll talk about birds of prey.
That's also fine. We are ready.
Okay. So thank you very much for the invitation. Actually, it's my first time in India. So I hope that when we call it a day, you will join us, me and Pankaj, to go to the city to party tonight. Because I didn't abandon my family for a week just to rest here. So we have to do something about it. Anyway, so I was told that the first talk should be some kind of a general overview of what we study. So I put together some slides from the lectures that I teach at the Faculty of Natural Sciences at Charles University. Then I realized that some slides were already presented by Helmut, so I apologize. I'll try to go quickly through them, but that's okay. So yeah, everything in life happens in cycles, as you will see. So this is actually one of the lectures that I teach. It's called Everything You Wanted to Know About Gene Expression, but Were Afraid to Ask. And it actually begin with this painting. Do you know what's the name of this painting and who painted it? Exactly. And who was the painter? Leonardo da Vinci. And I think it's actually a beautiful illustration of how close different religions and science are to each other. They are all trying to answer the painstaking questions that sometimes shatter us. With respect to what's the origin of life, where do we come from, what's the purpose of our life here, where do we go from here, and other things. And they, so religions provide some answers, science provides its own answer. And basically the goal is the same, at least I think. And the goal is to kind of mitigate our fears from the unknown, So to show us that we are not just a tiny little creatures totally lost in this huge universe, but there are some rules, and there is something to grab onto, to feel secure in this super exciting system that we all live in. And so I think this painting is a beautiful illustration of this closeness, because it's basically very religious painting. It's the core of Christianity. And this religious painting was painted by one of the greatest scientists of all times, Leonardo da Vinci. And so it just illustrates the marriage between his beliefs, at least I think, and his passion for science. And as you know, da Vinci was a master of secret codes and crypts. And so I think that this painting is also encrypted. And there's a message, there are hidden symbols that were meant to be relieved by our generations. So can you see them, these symbols? No? Okay, so there is double helix. See? There is nucleus. Cytoplasm, of course. And then here we have membranes with phospholipids, see? We have extracellular matrix in the back, so it's Mount Sinai most likely. And then most importantly, we have protein complexes, engaged in positive and negative interactions in this painting. Cytoskeleton over here. We have glucose and ATP on the table. There's one thing that he missed, at least I think. You know what it is? Exactly, RNA. Maybe it's hidden somewhere, I didn't find it, but maybe he didn't think of RNA. And yesterday, actually, we were told by Rajan that, we moved from the RNA-centric world to protein-centric world, and now we are coming back to the RNA-centric world, kind of, because 98% of our genome encodes RNA that it's not translated, and most likely it's composed of regulatory RNAs that at least co-regulate all these processes in cells with proteins. So I think RNAs are really, really super important. I'm surprised that the genius of Da Vinci really didn't think of them, but that's the way it is. Okay, so RNAs. Here we have the RNA in the central dogma of molecular biology. This is just to illustrate that I'll be talking about translation in my talk, in both of my talks. So translation of the message from RNA to proteins. So there are three key RNA molecules in translation process. I call them the holy trinity of translation: tRNAs, ribosome RNAs, and of course the mRNA. And we heard a lot about tRNAs yesterday, and I'll also talk about tRNAs in my second talk. So I'll just skip this, and of course about the charging. It was a beautiful talk. So just quickly illustrate the ribosome RNAs that form ribosome subunits, small subunit in blue, and this is a picture that comes from Venki Ramakrishnan. So I think Tanweer knows him. I really like it because it's super detailed. So you have small subunit formed of 18 ribosome RNA, large subunit formed of 3 ribosome RNAs, and you can beautifully see the mRNA decoding the T site, A site RNA with the decoded codon, P site tRNA, E site tRNA, which is about to leave. Here is the decoding center. This is the peptidyl center. Beautiful image. We can really see the core of this. And then mRNA. So I'm sure you know this, but when I ask my students, I'm quite surprised sometimes that they don't know. So would you know what's the major difference between mRNA in prokaryotes and eukaryotes? I'm not asking PIs, just the students. Pardon me? Yes, but there's even like really one shining sort of difference. Channel garden, of course. So the prokaryotic mRNAs, they start translation by, thanks to the C terminus of the 16S ribosome RNA, which base pairs with the short diagonal sequence, and this places the AUG codon or start codon directly into the P site, and that's different in eukaryotes, and they're also polycistronic. So in eukaryotes, it's different. You have, as you said, the cap and the poly A tail. And then, so the process is basically more complicated because the ribosome has to scan through this 5' UTR of the mRNA to locate the AUG. There are secondary structures that needs to be dissolved, unwind, and lots of regulatory features within these structures. And so the AUG is localized by the process called scanning. So now we'll proceed quickly to translational cycle. I'll just introduce some parts of it because they'll be important for the rest of my talk. And so this is, again, a copy-paste from Helmut's talk, actually. Ribosome is a molecular machine. And I'm sure that after listening to Helmut, you will know, what do we need to understand the function of a machine? Is it enough just to understand the individual parts? Or what do we know to understand how the machine works? We need to understand its functional states. It's a different engine. At least I picked a different engine than you did. It's 4-cylinder. Yeah. Okay, so the functional states. And this is, again, from Venki Ramakrishnan's lab. So these are the individual functional states of the translational cycle. So this is from bacteria. This all begins with initiation. In bacteria, we have only 3 initiation factors that cooperate to bring 4-methanyl tRNA to the small subunit. Maybe it carries alanine, I don't know, but at least we learned that it carries methionine. And so then we have subunit joining, and then it goes through elongation cycle that I will describe to you. Once there is a stop codon in the P site, the release factors come, they bind to the the A site, they trigger termination. We go through release cycle to recycling. So all these components have to be recycled to be able to start all over again with this initiation phase. So I'll introduce initiation phase a little more in detail because the second part of this talk will be important. And so it's initiation phase in eukaryotes, and because it's eukaryotes, I believe that eukaryotes were developed in EU, obviously. So let's take a look at this. It all starts with a simple thing, which is the methynyl tRNA binding to EIF2 to form the tRNA complex. For that, EIF2 has to be in a GTP bound state, which is ensured by the EIF2B subunit, which is an exchange factor charging EIF2 with GTP to form this tRNA complex. Then there's a bunch of initiation factors that interact with the tRNA complex. I want to focus your attention on EIF3 because this is the subject of the second part of this talk. They form a complex and they all bind to the ribosome to form something which is called 43S initiation complex. In this form, the ribosome is ready to accept the mRNA. The mRNA is capped. There is this 5' 7-methyl cap. It's recognized by the factors of the EIF4 group. EIF4E is the cap binding protein. There's poly A binding protein. So complex, a circular RNA. And so then these two assemblies, they come together and through bridging interaction between 4G and EIF3. And so this is called 48S initiation complex. And this complex is then ready to start searching for AUG, which is somewhere downstream. It has to go and inspect every single triplet to find it, basically. So this process is called scanning. It's very complicated, and this is one of the subjects of our study. So first, what has to happen is that, there's this is the interface side of the ribosome, and in the apostate, where you don't have anything bound, there's this little ledge which prevents mRNA loading. So this ledge, this is again from Venki Ramakrishnan, actually. So Laurie Passmore was published this 2007. So this ledge has to be dissolved first for the mRNA to load. That's important. So, and for this, we require two initiation factors, EIF1 and EIF1A, because then they rotate the head towards the body to allow the mRNA loading. When this happens, ribosome is basically ready to start scanning. This is just the illustration of this most important structural change. And so the ribosome is ready to start scanning. So there's lots of processes that I won't describe. Basically, it has to unbind the secondary structures. And as soon as the AUG gets into the A side, sorry, the P side, which is shown over here now. So there's few initiation factors that are released from the ribosome, and it triggers basically the reverse movement of the head towards the body, and this RS scanning. It's actually shown over here. So we have the anticodon of the methynyl tRNA. This is the AUG, and the ribosome scans, and as soon as the AUG gets to the P side, there's a base pairing, and this reverse switch, so the ledge is reformed. So in this way, the ribosome knows this is the right place where to begin, because it's, of course, super important for the ribosome to start translation at the right spot. And so we are now trying to understand how EIF3 contributes to this process in mammalian cells, as you will see in a few minutes. And then we have subunit joining, which is mediated by EIF5B. So this leads to eviction of some of the remaining initiation factors. And then there's GDP hydrolysis on EIF5B. EIF5B monitors intersubunit bridges between both subunits. And when it's happy with the conformational sort of arrangements, so it triggers the GDP hydrolysis and leaves the ribosome. So the initiation ribosome is now basically ready for elongation. So the elongation phase, very quickly, because it's not relevant. So there are these functional states that I described at the beginning. So this is the paper from Christian Span from 2015 that was published in Cell. And so he put together these, collected particles to show us all individual states that are, in a sequence going through. So the ribosome goes through in the sequence during elongation. Now, as I told you when I was asking Helmut, so now I think he has like 30 of them. So it's even more complicated and more detailed. So the first thing, what happens is that the tRNA complex, which is composed of elongation factor 1A bound to tRNA and GTP, it comes with the tRNA. The tRNA is, with its anticodon, is sampling the codon. So this process is called codon sampling. And it's fascinating, at least to me. It will be always fascinating. This is again from Venki Ramakrishnan. So it just depends. The sampling depends on these two bases. These two. This is from bacteria. These two adenines, and they actually monitor a base pairing between the codon. So I'm sorry, this is the empty A site. Over here, you can see in the P site, the tRNA base paired with the codon of the mRNA. And then when into this space, the A site tRNA comes. So when there is a perfect base pairing established, so there's a special geometry of this RNA, RNA mini helix, which is formed. And this mini helix is very important because its geometry is actually monitored. So not the bases are monitored, but the geometry is monitored by these two bases. And if the geometry is accepted as the correct one, then the tRNA is accepted. And so this actually, the fact that these bases monitor geometry and not the bases, not the base pairing, explains the fact that you can have different modifications. You can have wobble base pairing, you can have different, like unusual pairings, because as long as these modifications sort of fix improper geometry to establish the proper geometry, then these bases are happy and the tRNA can be accepted. So after this codon sampling, this actually, when the base pair is correct, this leads to GTP hydrolysis on elongation factor 1A, and then we go through proofreading step. It's basically the second step that where ribosome can reject the tRNA, and during this accommodation step, which is then accomplished by basically moving of the amino acid on the CCA end of the tRNA towards the PTC, the peptidyl-transferase center, as you can see over here. So at this stage, basically, the peptidyl-transferase reaction can occur. So the process becomes irreversible. And so the polypeptide chain, which was previously on the P side tRNA, is now through this peptide formation attached, moved to the A side tRNA. So at this stage, what we have to do, what the ribosome has to do, it has to basically move this tRNA from the A side to the P side to allow the next tRNA to come. So this is called translocation. Translocation is facilitated by elongation factor EEF2. I'll show you in a second how this works. And this is basically the key step of the whole process. It has to move the mRNA by 3 bases so that now we have a new codon in the A site for the new tRNA to come. And it's a quite nice actual illustration how this works, because so you have large subunit, you have small subunit, and this is actually the state at which the ribosome is after peptide formation. So we have the A side tRNA, P side tRNA, E side is empty, and there's spontaneous movement of the ribosome, the small subunit towards the large subunit, going back and forth. And what has to happen, actually, we need to get these tRNAs to P and E side on both subunits. So it shifts back and forth like this. And to keep these tRNAs in the P and E side also on the small subunit, we need this factor, we need EF2G, which comes there, basically prevents this back translocation of the amino acids to end up in this state. And actually, it's quite nicely illustrated with this Brownian ratchet machine. So this is a spontaneous movement back and forth. It's called ratchet movement. And we need the pole, which is actually this EIF2, which comes and blocks this backward movement of the tRNA so that the A site is then empty and ready for the next cycle. And so this is just a marching of tRNAs through the ribosome. I think you have also had a similar slide on your presentation. Anyway, so at this stage, then, we can accept, so the ribosome can accept new ternary complex with new amino acid, and the whole cycle repeats itself until the A site, until the stop codon gets into the A site. This is just very quickly, these are, functional states of the ribosome that actually depict what, like real structures that depict what I was trying to show you with these cartoons. So we can see we have empty A site. This is from my friend in our country, Gabriel Demo, who got these papers published recently. So now, the A site, the tRNA comes. So this is the stage of the codon sampling. So you can see that the anticodon now becomes properly base paired, so there's a perfect match. So this triggers GTP hydrolysis, which leads to ejection of GTP-bound elongation factor 1A. So in this case, it's actually EFTU because it's bacteria. And then there is this accommodation proofreading. So the CCA end swings towards the peptidyltransferase side to start kissing, the P site, CCA and tRNA. And then there is peptidyltransferase, reaction. And so we have this, translocation. We have this, the polypeptide moves from the P site tRNA to A site tRNA, and then it's ready for translocation. I don't have slides for this because that would take forever. Oh, so then we have a termination phase. So for termination phase, of course, we need to get, stop codon. The ribosome needs to get the stop codon in the A site. So as soon as there is, stop codon, so there's the tRNAs cannot bind, at least not with high frequency. So what is occurring is that we have release vectors in eukaryotes. They are called release vector 1 and release vector 3. Release vector 3 comes GDPA, so it comes with bound GDP. So it binds to this through this Nix motif to the AUG, which mimics, the anticodon of the tRNA. And then again, as soon as this is recognized as a proper stop codon, there's GDPA hydrolysis on the release vector 3, which leads to its ejection from the ribosome. And then there's another factor called ABC1, which is ATPase, actually NTPase. It binds in the place of release vector 3 and promotes conformational change, which leads to the movement of this GGQ motif, which is required for hydrolysis of the peptidyl tRNA in the P site to the peptidyltransferase center. And then there's a hydrolysis of this bond, recently challenged by, I forgot the name, the science paper, how this works without water. And so after this, we finally have, the matured protein, which is already assembled, goes to the cytoplasm to work, and the ribosome in this post-termination state. So in this post-termination state, ribosome has to be recycled. So as a recycling factor, we require this ABC1, because now the ABC1 is using actually ATP to create mechanical chemical force, which splits, basically rips apart the subunits. And when this happens, there are a couple other factors, like some initiation factors, or this dedicated factor EF2D that binds to the small subunit and dissociates tRNAs from the E side and P side, and also the mRNA. And that's the end of the cycle, basically. And now, if I have time, I will go to the first story of mine. Okay. So, and that's something which concerns this first part. So how AUG is selected as an initiation codon, and what factors are required, how this is all coordinated. And so I'll just begin with a brief introduction what the EI3 is. So as I mentioned, EI3 is one of the key initiation factors. It was shown to promote nearly all initiation steps, and especially in yeast, because in yeast, there was a really lot of work being done by Alan Hinnebusch, Tom Dever, John Lorsch, Alan Pankaj over there as well, my lab, other labs. And so it was also shown that it's a critical structural scaffold, because it's huge, it's a cellular subunit, so it binds to the ribosome, and it helps to other factors also to bind to form this 43S conjugation complex, which is required for mRNA loading. Besides that, it was also shown it can have special functions, so it can bind to the 5' cap and regulate mRNA translation in gene-specific manner. So it's required for bulk translation, but also for specific translation of specific mRNAs under specific conditions. Everything is specific, of course. And so its dysregulation was shown to be linked with oncogenesis. This is also one line of our research that I'll not talk about today. And finally, it doesn't work only in translation initiation. Surprisingly, it also controls termination, stop codon read-through, which I'll talk about in the other presentation. So it's a very pleiotropic factor being involved in many things. But today, and also it promotes recycling and reinitiation after the translation of short open reading frames. So when you have a mRNA where you have the main ORF, then you have short ORF which precedes. So the short ORF normally should be translated and then lead to dissociation. So the main ORF would never be translated. So EIF3 and other factors are there to allow, so when the short ORF is translated, these factors take care of the ribosome to stay on the same mRNA, continue scanning, and also translating the main ORF. And this is, of course, a highly regulated process. So it's also involved in there. But today, I will focus only at this step. So how EIF3 coordinates the process of AUG start codon selection. So this is a summary of, as I said, many publications from different labs, including our Alan Hinnebusch was, I think, my former supervisor was particularly stubborn in trying to understand this. He would never give up and on anything. So this process, it has three key players that are required. So it's, of course, the EIF2, which comes bound with the ternary complex, as a ternary complex with a methyl tRNA. And then there are two factors, EIF1, EIF5, and EIF1A, which is not on this picture. So these four players are super critical for this process. And so we have shown that, in yeast, all these three players, not EIF1A, but EIF2, EIF1, and EIF5, they actually interact with EIF3. This is a very naive sausage model that we put together, I think, in 2002 or something like this. I still keep it. I mean, so far, nobody has solved the structure, of free EIF3 outside of the ribosome. So this is the best structure available, correct, Tanweer? No? Okay. Anyway, so this, complex interacts with EIF1, as you can see here over there. It also interacts with the EIF5 and with the ternary complex. And it does so through two subunits, EIF3A and EIF3C. And we have proposed and showed some genetic data that these interactions with these two factors are actually critical for coordinating these events. But this is in yeast, right? So what about in humans? There's very little known how this process is regulated in humans. And you can also appreciate that yeast EIF3 has only 5 subunits, as well as opposed to human EIF3 has 12 subunits. It's much bigger, a lot more complex. So, we have done a lot of experiments, and again, not only in my lab, to show, what's the composition of human EIF3, how it assembles, what's the functions of individual subunits, blah, 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 blah. So, but the major focus of my lab now is to understand how, it helps to recognize the AUG, and it's a giant void. Nobody knows. Nobody knows how this actually works. So we are trying to fill this giant void up now. So we started by very simple, classical biochemical approach. We took all EIF3 subunits, we fused them to GST, we, radiolabeled, synthesized radiolabeled proteins, EIF1, EIF5, EIF2, all 3 subunits of EIF2, and we just tested the protein-protein interactions in vivo to see if they correspond to what we have seen before in yeast. And we could show that clearly that EIF3C, as in yeast, interacts strongly with EIF1 and also with EIF5. Fine, so we felt confident, it's conserved, let's move on. So then we, together with Yasser Hashem, actually we were able to get the structure of the ribosome from trypanosomes, and we could actually show that we could not see the context, but we could show that the N-terminus of EIF3C, which is over here, is in close contact with EIF5, should be binding somewhere, and also, so this is the N-terminus of EIF3C, is also in close contact with EIF1. I believe I'm almost blind. To the right factors, yeah, it's EIF1. So even in the structure, they seem to be close, which fits with the biochemical data. So then we went on to map, the precise binding sites, and so same approach, GST pull downs basically, and so we could actually show that the first 30 amino acids of EIF3C in mammals is sufficient and required for binding to EIF5, I'm sorry. And then that the region between 171 and 240 is required for binding to EF1. So that's why I call this presentation EF3 at the tip of the process, because EF5 is actually binding to the very, very tip of EF3C. So we had this, lined out, and then, we were curious to see if we can confirm this data by some in vivo approach, because this was all in vitro. So we did co-IPs. We used the antibody against EF5, which is highly specific. So we pulled out the wild-type complex, and then we used the different knockdowns that we have established over the years to see if we knock down individual subunits, and we know exactly what happens if you knock different subunits down. So if we can still pull down EF3 through EF5. So for example, this is EF3K knockdown. So K is here. And we know that when we knock down EF3K, we lose also L. So then the left leg is gone. Is it left? Yes, its left leg is gone. But EF3C is still present. So that shouldn't affect the pull down, and it didn't. So you only see that L and K are missing. Then we knock down E, which is this guy. So when we knock down E, we lose basically the entire, half of this complex. But EF3 is still present in most of the complexes. So we see some impairment, but it's still binding. And as long as this is EF3C knockdown, as long as we get rid of the EF3C, so this is what remains, and then the antibodies are powerless. We don't pull down the rest of the complex. So this was in vivo confirmation that EF3C is really bridging this contact with EF3 in cytoplasm. Unfortunately, still with just the knockdowns, so that disintegrate most of the EF3 complex. Anyway, so with this in hand, we finally started to ask the question, so is it important for AUG recognition in humans? For this, we have something which we call the SUI assay, and that's very simple. So you have just a luciferase. The luciferase is beginning, and the initiation code is either ATG or near-cognates as a CUG or GUG or UUG. And then you can actually do the ratio of the, let's say CUG initiated luciferase versus AUG initiated. You normalize it to the mRNA levels after transfection, and you can calculate these ratios and plot them. So as you can see, when we knock down ER3C, so we significantly increase GUG or UUG or AAG ratio with respect to AUG, which means that we really have impaired AUG selection process in humans. When we do this with K or E, so this in the case of E, we see some increases, but they're not significant. With K, we barely see anything. Just with this one, just tiny. So this was indication that yes, missing C may have a role, may impact the AUG selection process in cells. But the obvious caveat is that we are not working with the whole complex. The complex has fallen apart. So this is interesting, but of course not enough. So then we said, okay, let's try to look into at the sequence if we can find out the residues that could be mediating these contacts, and by mutating these residues, hopefully we'll be able to get single point mutations, and with these, if we CRISPR them in, we can get a better picture with the intact ER3. So this is just again the same approach, and to cut the long story short, so this arginine and these two phenylalanines are the key. If you mutate them one by one, they reduce binding. If you mutate all three of them, the binding is gone, even if you use the full-length protein. So these three residues are absolutely essential. So it's a really very tip of ER3C, essential for bridging the interaction with ER5. Okay, so let's go CRISPR. So we designed the CRISPR analysis for these three single substitutions as well as for the triple. But the problem is that on chromosome 16, ER3C gene has 2 copies plus 1 pseudogene. In diploid cells, we have 6 alleles, including the pseudogene, that we have to hit if you want to get the homozygous mutants. So 6 alleles to target to obtain exactly the same single point substitution. I thought this was impossible, but I was actually proven wrong by my student who tried it, and we actually succeeded with exception of the F4A. So we have now single substitutions for these two homozygous clones, several of them, and we have two homozygous clones also for the triple. So it worked nicely. And now we can really look into the role of these of the ER3C in initiation in AUG selection, because we are just, you know, I'm almost done. So we are hitting only the specific interaction. So we are losing binding to 5, but the rest of the complex is intact. So, and that's the proof. So when we use anti-flag antibodies against ER3C in the system with the triple mutant, we have perfect binding to EF1, it's intact, but the EF5 is gone. So it really works. So this is from the CRISPR, you know, this is from the triple mutant, but we see the similar thing with even in the singles. So then we could really look into the consequences. So this is the viability assay. This is the wild type curve. So all the CRISPR clones, they grow a little slower. They don't like it. Polysome profiles, if you are familiar with, they give you an approximation of the efficiency of initiation process in cells. And actually, you can see that they are, especially for the triple. So what you measure is the amount of heavy polysomes. This is the signal of really efficient translation. But if this goes down and this ADS monosome goes up, as in this case, that's a signal of impaired translation. That's what we see. And then we went to the SUI assay, actually, you know, this assay that can monitor specifically the relax initiation at these neocognate codons. I have just this picture, but we have a lot more. So actually, for these singles, we can show nicely that also with the GUG and UUG, we have increased we have relaxed initiation. So it really impairs this function. And then this last thing to show is this competition assay, which I was really happy when I saw this. So we overexpressed only the N-terminal part, the first 180 amino acids of ER3C, hoping that we'll be able to, you know, this little piece of protein will be able, peptide, will be able to compete with wild type ER3 for ER5. And why? Because it was actually shown that when you overexpress ER5 in cells, it also increases this neocognate versus AUG ratio in cells, because it also relaxes selection, which is shown over here. So this is basically the cells where nothing is overexpressed. When you overexpress ER5, then the UUG to AUG ratio increases, as you can see over here. And so we are thinking, if we overexpress the wild type fragment, which can bind to ER5, so this wild type fragment could sequester ER5. So this excess amount would be basically lowered, and we should reduce this ratio. And we did quite nicely. And then when we use the mutants, either single per mutants or triple mutation, we are back to the original state. So they cannot bind ER5, so they cannot sequester it, which means that ER5 can again act as this hyperactivator and so trigger AUG selection, even if there is no AUG, but there is just a neocognate AUG. We have a lot more, but I don't have time to show these things. Just to summarize this part, so the human ER3, we could show that it interacts with ERF1 and 5 through its N-terminus, as in yeast. ER5 contact is at the very tip. And then when we disrupt this contact specifically with the CRISPR, you know, integrated mutations, introduce mutations, so we lose fitness. I mean, not we, but cells lose fitness, and we increase initiation at neocognate codons. So this actually implicates that ER3 can fine-tune the AUG selection process in mammalian cells. And now we have mutations even in ERF1. We are doing single molecule experiments with Jody Puglisi in Stanford to, you know, get the more close look. We are doing ribosome profiling experiments to show how these mutations affect initiation, you know, selection of the AUG codons or neocognate codons on the whole genome level, all sorts of things to be able to come up with a nice, convincing story. So this is just an acknowledgement slide for the first presentation. Okay. So this work was started, and all the CRISPR, you know, magic was done by Terka from my lab. Now Anya took over, and then Pragya is working on EF1 story now, and Philip is the major force of all of this work at the moment. These are the funding bodies, and I think I'm done with the first presentation. Thank you.
We have time for a few questions.
EIF5 also has two domains, NTD and CTD. So which of the domain interacts with EIF3C?
So EIF3, EIF5 NTD, as far as I know, has not been shown to bind to anything specifically, because this is the domain that's supposed to trigger with the Argentine finger, the GDP hydrolysis on EIF2. So all proteins, and there are many of them, surprisingly, all proteins were shown to interact with the EIF5 CTD. So we are mapping the binding site on EIF3 CTD. There are some hints from the literature, and it's definitely also the C-terminal part of EIF5.
I have another question if there's time. Yeah. So EIF3 also occurs in subcomplexes, at least in yeast it has been shown. So, and these subcomplexes are able to recruit mRNA onto the ribosomes. So, like, do you have any?
If I have what? Yeah, you are referring to work of Colin Shivaya, I suppose, right?
Yes.
That, I mean, Colin
They've
is
shown by mass photometry that EIF3 occurs in subcomplexes.
Yes, so this was shown for mammalian EIF3, and we have shown this as well when we disintegrate EIF3. I'm not so sure, how physiological this is. I think in a normal cell, the subcomplexes will be in minority, because what we also have shown is that EIF3 actually forms, assembles, like there's a co-assembly during translation of different subunits, so it co-assembles. So I'm not so sure how many of the subcomplexes would be functional and how prevalent they would be in the cell, but it's possible, and they may have different roles. In yeast, I like Colin a lot. He's my great friend, and I told him several times, I think what he has shown is not physiological, So they can occur, but if they have a real role in the cell, I personally doubt it. But that doesn't mean that it's not true.
So this interaction between 5 and 3, does it have a role to play in, even for the the hydrolysis of GTP in EF2?
Indirect
scanning.
Yeah, so indirect. What we are trying to show with Jody, actually, you know this last paper that was published just a couple of months ago in SMB. So they actually show that in mammalian cells, EIF5 is not a part of the scanning complex. It comes as soon as the AUG is already in the P site. And then there's this interplay between, because there's a mutual exclusive binding between 5 and 1, and so the 5 needs to come to replace 1 to trigger the hydrolysis. And what we think and what we are trying to show, and we already have some indications, is that EIF3C is coordinating this replacement. So 5, they both bind, and EIF3C is needed for like a fine-tuning of this switch between 5 binding to one place for hydrolysis to occur through the NTD of EIF5. That's the working hypothesis.
Yeah, but then 5 comes when the start codon AUG is at the P site.
Yeah.
But if you look at the older data from Hinnebusch lab, that
But this is yeast, So in yeast, I agree. John showed quite convincingly that EIF5 is part of the complex. So it can be different in yeast. I don't know. There are data supporting it. There are data disputing it. I personally has always believed that it's true. But I think what Jody now published, seems to be quite convincing that EIF5 is not part of the scanning complex, and it comes at the stage where AUG is already in the P site.
So has anyone looked at the mRNAs where there is a stem loop structure little downstream of AUG that actually sort of compromises the selection of AUG also? And so how will these ligands behave in that kind of report?
So we are checking the Cossack sequence, of course, the Cossack context. So, and they are also having an effect. So it's with the medium Cossack, they are more relaxed again. With respect to secondary structures, so secondary structures should slow down the whole process. So if they are placed in the right position, so that the AUG, so the dwelling time of the ribosome in AUG or near-cognate codon is increased, then of course they by themselves they would increase initiation at the near-cognate codons, and in a way, yeah, we haven't thought of it, but maybe it's another thing to try. So that should be additive in principle. So the stem loop combined with these mutations should generate additive effect, so even more relaxed.
Yosh,
I'm worried now.
yeah, looks like I mean, we have some data which is a little bit contradictory. What you are... Hello. So what I'm wondering that 5 has to be there, what Tanweer has said, 5 has to be there to trigger the GTPase reaction. So if 5 is not there because of its interaction with 3C, probably then you should not see the Sui phenotype.
I'm not saying it's required. I think it accelerates this process as far as it's like.
How? It cannot, because it needs the GTPase.
You should know, you are studying EF5,
Yeah, that's what I'm saying. Like our data is a little bit different, but anyway.
But you so this is mammals, we don't, and human cells. So it could be really different in yeast. Yeah, I mean, we can talk about it later, but I'm not really, we are not going into the role of EF5 in particular, So we are just trying to show this coordination process, how this is happening. Yeah. But there might be a major difference between yeast and humans, that's for sure.
So is it like, do you think it may be an inhibitory thing for a short period of time? It is inhibiting the interaction?
You mean the mutations?
No, the interaction of 5 with 3C would be an inhibitory in nature.
In a way, yes. What we actually think and what the data suggested I didn't have time to show is that EF5 has to be kept under control, And so if it doesn't bind to EF3, it just acts spontaneously and prematurely. So it's like hyperactive in a way. So if it binds to EF3C, then the whole process is coordinated because EF3C makes sure EF5 doesn't fire too early. That's what the data that I didn't show seems to indicate, but they are preliminary.
So, just wanted to know, what do you think about the evolution of EF3, from yeast to humans, from 5 to 6 subunits to 13 subunits?
Yeah, I think yeast come from different planets. There's a model system. Because, Pombi has 9 or 10 subunits. So it's yeast is budding yeast is fantastic model system, but in many ways, especially for this, it's completely wrong because it doesn't resemble even the closely related yeast. So, there are lots of stories, ideas and theories about why it has only 3 subunits, and I personally don't have any strong opinion which one is true.
Much of the EF3 is actually kind of on the surface.
Oh, you mean of the ribosome?
Yeah.
Yes, but it has two arms that seem to embrace. That's actually his work.
So I would suspect that this would also provide a way of regulation by so many other factors which can sort of interact at that place. Because otherwise, why would you have so many subunits of EF3?
Oh, yes. I mean, so if we don't talk about ER3C, I didn't maybe emphasize that ER3C is actually not part of this bulk of ER3 on the back. It's an arm which comes from the left side and reaches out to the P side of the ribosome on the other side.
Yeah, so I was kind of extending on that because, different subunits and during evolution and all that.
Yeah. So that's why we have started this, so many subunits, what for? So we have some ideas, for example, K and L, they might promote mRNA loading, for example, but at the same time, if you knock them out, even if they're not essential. So if you eliminate them, the translation rate goes up. So they seem to be inhibitory. EF3D is super important for pretty much everything. It can bind the cap as well as EF3L can bind the cap. And so most likely they will be involved in these specialized cases. So for the bulk of translation, this is what we also showed. It's enough to have these 5 subunits in principle, even in mammalian cells in vitro. But for if you go with the cell through regulation and different stresses and stuff like that, then you might need these subunits to interact with specific mRNAs. So either suppress, as Jamie Kate showed, or induce their translation. So most likely regulatory purposes. Yeah.
Any more questions? Okay, can go to the next part.
Tea break or something? Fine.
No, no.
Yeah.
We continue.
Okay. So do you mind if I have tea break?
Yeah.
No. I've voted him. I was just trying to. Yes, I'll just drink a little bit of water. So this is another image that I like to show. It basically, it's the body of a guitar, and so it looks like a ribosome, 60th subunit, 40th subunit. I kind of perceive translation as the orchestra. So all these tRNAs are like a notes that have to they count the chords, and the ribosome has to play them well. If it doesn't play them well, cell would know. So, and that's why we study this process of decoding and regulation of decoding, because it's extremely critical for the cell to play all the tunes well, for all the processes to work nicely. So initiation, elongation, we have gone through this, termination as well, but there are cases. What?
Recycling.
Not recycling. I'll be talking about, maybe if it works, yes, about the read-through. So in some cases, when the stop codon is in the A side, termination can be sort of ignored because we have tRNAs that are necognate to the stop codon. So basically they have 2 out of 3 base pairs, and then they can prevail and they can actually bind to the A side and allow read-through. So the translation goes on, it doesn't stop here. It goes on and it actually creates protein with the C-terminal extension. And the C-terminal extension can have a specific sort of function. So it can change the role of the protein in the cell, either different localization or even different function. So it's either spontaneous, which occurs with very low frequency. This is basically an error. Or it is programmed, so there are specific features that can stimulate it to make it programmed, so that this means that cell really requires that a certain proportion of proteins is extended to get to let them do what they need to do. Yeah, and so this part of the story will be the talk will be about the read-through, and the reason actually I call this talk "Starts Worth 0 Pence and Stops Making Sense" is because, with all this initiation sort of work we have been doing since, I don't know, 1997, we have published a lot of papers, and maybe interesting, some of them are, but we have never, come up with a real breakthrough. And I think every scientist is dreaming about some breakthrough article that the people will remember at least for a year and maybe more, and not just for a few weeks. And so what I would just want to say that, with starts, we didn't really succeed, but with stops, we finally got the breakthrough. So the breakthrough for our lab came with the read-through. So just quick history lesson. So till, as I said, something like 2012, this is the review that on initiation, we were focused on initiation, but we were aware of the paper from Tatiana Pestova, actually from, I think, 2010, where she, in the reconstituted system, she could show that initiation factors, including ER3, are critically important for the recycling step, which is on the other side of translation, not initiation, but basically termination, recycling. This was actually a mammalian rabbit reticulocyte lysate system, purified factors from retics and then reconstituted. So different organism and in vitro. So we thought, okay, so we know what to do with yeast. Let's try to show that this is also true in vivo. And so we thought, so okay, so some assay, but there were no assays available. So what can we try? So the only approximation that we could think of was just a simple dual luciferase assay that monitors stop codon read-through. This is as close as we could get. And so, you know, we had zillions of mutations in EIF3. So we said, okay, so let's test these mutations in this dual luciferase reporter to see if they impact read-through. If they impact read-through, maybe that means that they may also really impact recycling. That's the image that's 15 years old. And so the gray bars are wild types. I mean, the dark gray, the light gray are the mutants. And so we were surprised that most of the mutations that we have in individual EIF3 subunits, actually, when we use these mutations, they improved fidelity of read-through. So all of a sudden, the recognition of the stop codon was more, by Reese factor, was more precise, which was super surprising. The only exception was the associated factor, which binds to EIF3. It's called HCR1 in yeast, and this actually relaxed this stop codon selection. So that was encouraging. Okay, fine. So we did a lot of experiments, thinking of the breakthrough finally. So we tried Nature, Cell Science, you name it. It was reviewed, rejected almost everywhere. So finally we got it published in PLOS Genetics. Quite nice. Nobody believed us. Now it's one of the most cited papers coming from our lab. But that doesn't matter. It's just to illustrate that you don't give up. Just go, and the future will show. And so we just show that these two factors, EI3 and HCR1, are controlling translation termination, even though they should not be there at the ribosome. So it means that they bind to elongating ribosome, also terminating ribosome. This is one surprise. And by binding to terminating ribosome, they promote translation termination control, and they promote stop codon read-through. Then we went even further. And so, we were thinking, since it promotes translation stop codon read-through, so, does it do it, EI3, does it do it for all tRNAs or only for some tRNAs, and how does it do it? So a couple of years later, we published this paper where we actually could show that for some tRNAs, but only some tRNAs, EI3 is really instrumental in promoting read-through, and it does so by reducing the termination activity of EIF1. And so this was quite nice. Then if you look, so this is the key assay that I have to describe because I'll be talking about it most of my talk. So the dual luciferase assay works in the way that you have two luciferases, you have Renilla and Firefly. In between, there is a cassette, in-frame cassette, where you can actually place a stop codon in different contexts. And then you have also a coding codon as a control. And so what you actually measure is ratio of Firefly to Renilla with the stop codon normalized to this sense codon. And also you have to monitor the levels of the mRNA. And so you get this percentage of read-through. So for example, for this context, you have some percentage. If you use specific program context, you can get much higher read-through with these specific contexts. And the way we approach this this question, does EIF3 promote only some incorporation of only some tRNAs, was by adding one more step to this. So my student, postdoc later, developed this assay. She simply overexpressed different tRNAs in this together with the dual luciferase assay. And she was asking, so by overexpressing these tRNAs, do we see an increase in the ratio of Firefly to Renilla or not? And you can see here it works nicely for those tRNAs that are efficient. So like here you have empty vectors, so nothing is overexpressed. If you overexpress tyrosine tRNA, you increase 8-fold the read-through, the glutamine tRNA 13-fold. So this works. It doesn't work with all, of course, only with few. And so when we publish this paper on EI3 and which tRNAs it promotes to bind to the termination codon. So Petra looked at this table and she said, look, so there are 9 tRNAs that are near cognate to stop codons. So I would like to test them all. I would like to see if all of these tRNAs, all the isoacceptors, which means they have different anticodons, but they carry the same amino acid, or isodecoders, so they are the same anticodon, same amino acid, but just differences in the backbone. So if with all these tRNAs, if they promote or don't promote, and I said, this is crazy, it's tedious, a lot of RNAs, and this is, we cannot publish this because it'll be so boring for everyone to read this. But she didn't care, she totally ignored me. She developed this assay where she could test all these tRNAs in large scale in principle. And then she came up from this list with 4 tRNAs that in her hands, and only some isoacceptors, as you will see, in her hands were really, really super efficient in promoting read-through. And as you can see, she was fairly productive and successful. So I'm quite glad that she didn't listen to me when I said, don't do this, this will be boring. Anyway, so don't listen to your PIs sometimes, it makes sense. And then, and of course, then you will feel a gratitude when they come to you and say, oh yeah, you were right, I'm sorry, I was wrong completely. Anyway, so, and luckily for us, in the meantime, around the same time, so Olivier Name's lab from France and Alan Jacobson's lab from US, they used different approaches that they came up with the same set of tRNAs. So these 4 tRNAs, the tyrosine, glutamine, tryptophan, and cysteine tRNAs seems to be really, really super efficient. And we call them read-through inducing tRNAs because they are efficient a lot more than all the other neocoordinate tRNAs. So here they are. So this is the Petra's contribution, which was huge to this. So we have these tRNAs. In the case of tyrosine, we have 2 isoacceptors. I mean, the tyrosine tRNA can read both UAA and UAG codons. And for the rest of the talk, I will just focus attention on this GLIN tRNA. The GLIN tRNA is actually, it comes in yeast, it comes in 2 isoacceptors. It's either CUG or UUG. And when Petra did this large-scale screen, she actually noticed one interesting thing, and that is that the CUG isoacceptor promotes very efficient read-through, but the UUG isoacceptor doesn't. And so we were kind of puzzled why, why is this difference? So I thought this is finally interesting for me, because there must be some mechanism, how the ribosome distinguishes these 2 tRNAs. Why is it so? But it was not interesting for her at all. So she said, I won't do this. So, I had to hire a new PhD student, naive, who would not tell me no. And Zuzka, who's actually, also super talented. And so she said, okay, I'll do it. Petra stepped out. And so first thing she asked was, is it because of the, the anticodon identity? So in other words, so if we swap these anticodons, if we'll swap the outcome, the answer was no. So we tried modifications, other things, nothing. So it was not about the identity of the anticodon. It was not about the modification that we could think of. So when we compare the backbone sequence of these 2 tRNAs, we realized there were only these 3 differences. And the most important difference is this A in position 42, which is in this B isoacceptor, and in L isoacceptor, and in all other isoacceptors, you have actually G. So we asked the question, so is it this? Is it this base pair in the anticodon stem that makes the difference? And the answer was yes. Because when we mutated this, when we basically swapped A with G, we completely killed this activity, and we got to the level of the L isoacceptor. So then we did it in a reciprocal way. We tried many more mutations. So to cut the long story short, so for this tRNA, it appears to be important. We call it the 4 base pair rule. So it appears to be important that at this position, at the 4th position of the stem, there must be pyrimidine purine base pairing. It can be, any of the pyrimidines, any of the purines, but it must be Watson-Crick base pairing, and must be pyrimidine at position 28 and purine at position 42. Otherwise, it doesn't work. Otherwise, you completely kill the activity. And luckily, this is conserved in T. brucei, where we tested it in collaboration with Zdeněk Parda. And so when I saw this, I was like, I was right, cool. So we have, some nice mechanism, a couple more experiments, and we can publish it in some nice journal, hopefully soon. But then we were hit by the lightning because, actually we had to hit the brakes because I was contacted by these 2 guys from my country Jan Motýpka and Julius Lukács, and they told me, Leos, you work on read-through, yes. So you have to come to our talk because we have something which is super exciting, and I'm sure you will like it, and maybe you will help us to solve the puzzle. So they actually catched. So these guys, they travel the world, and they carried microscopes on their in their backpacks, and they go to the nature. They collect different bugs, and they isolate different parasites, mostly trypanosomatids. And then so in the evening, they go to a restaurant, they eat, they drink, and then they set up the microscopes and they dissect these bugs, and they collect the trypanosomes, and then they smuggle them back home. Seriously, they smuggle them back home. They don't report anything. And then they sequence them, and they're looking for particular interesting things that could, because the trypanosomatids are super crazy. They do everything in a different way than we do. They're trying to come up with some interesting sort of features that by which these organisms differ from from all other eukaryotes. And they caught this bug, and they isolated this trypanosomatid, which is called Bastochydia nonstop. You will see why it's called nonstop in a second. And they told me, so let's take a look at it. And I was not convinced that it's really worth pursuing. So I said, no, I'm not. I mean, we are busy. We don't have any any some free force to work on this. And they say, you have to come because this bug actually lives at your doorstep. So this is the place where I live, and it's like 15 kilometers. So they caught this bug 15 kilometers from my house. I said, okay, so I'm a patriot, I will come. Let's see what we can do. So I came to the talk, and they actually presented something which I need the genetic code for you to start with explaining, to be able to explain it to you. So you all know the genetic code, basically, 64 codons, 61 sense codons encoding 20 amino acids, and then 3 stop codons. So when it was discovered, it was believed to be universal, frozen in time. We know it's no exception. So UAU would always encode asparagine and so on and so forth. But with time, it was shown that there are at least 50 different alterations of the genetic code. You can have different changes of the sense, either from sense to sense, different sense, or from sense to stops, from stops to sense. So it completely rewires the genetic code. I've shown you that with these stop codon read-through tRNAs, this can also, in some cases, change the sense from stop to sense codons. So it's not frozen, actually. It's not universal. It's quasi-universal. You can think of it as frozen in time with the global warming episodes. And so basically, this is the list of all alterations. No, not all now, because this is from 2016, but from all alterations that were known at this time of the genetic code. And I just want to draw your attention to these 3 organisms. So Blastocrytidia that I mentioned, and also this ciliate, Condylostoma pardusia, and this dinoflagellate Amobafria. Because in these organisms, around the same time, 2016, this one was 2019, it was shown that these organisms, they have all 3 stop codons reassigned in the nuclear genome. All 3 stop codons reassigned in the nuclear genome. So what does it mean? They are basically non-stop. So how does it look if you have stop codons reassigned? So actually, if you imagine a gene as a sentence, beginning with a capital letter and ending with a period, and a genome as a book telling an entire story, then thanks to the capital letters and the periods, we know exactly what the sentence, when we read it, what the sentence begins and where it ends, and the whole story makes sense. What happened in these organisms is actually that the extra periods infiltrated the sentences. So we no longer know where they really end. It becomes a mess, and the whole story becomes disjointed. So you can see it's really hard to read this, so you don't know where the end finishes. So this is the genome of these organisms. This is how it looks. And in our terms, so for example, if you look at one of the genes, and the sequence, so this is AUG, and then you have, in some part, you have 3 stop codons, in frame, one after the other, and then this is the genuine stop codon. So most of the genes look like this. They have so many stop codons in their genomes, yet they still live. So in 2016, Marek Eliash's lab actually predicted that the UAG and UAA are encoded by glutamate and tryptophan, and UGA is recoded by tryptophan. So we started by doing experiments, mass spec experiments to prove this prediction, and actually they were right. Yes, so glutamates and tryptophan that recode these subcodons. But of course, the main question was, how are these in-frame subcodons in the coding regions recoded? So how this organism can actually survive? So very quick answer for the glutamate. So for UAN and UAG, so these organisms, they have new genes. They actually come from the original glutamate RNAs, and they mutated the anticodon. So super simple solution. So UAA now has a perfectly complementary tRNA, new tRNA, glutamate tRNA. So it base pairs with the anticodon, and the same is true for UAG. So it's a very simple solution. But what about the UGA? So in UGA, with UGA, we could not find any tRNA that would have this perfect mutation that would make tryptophan tRNA fully cognate to UGA. So there must be a different mechanism. What is it? So we checked the sequence, and there were some differences, but what was striking for us was that this anticodon stem of this tryptophan tRNA in this organism was actually shortened, not with respect to the length of the whole tRNA, but just the base pairing was not formed by 5 base pair, as it is in most of all of the eukaryotes, but the stem is formed only for 4 base pairs. So the top base pair was unpinned, and so we thought maybe this is the mechanism by which these guys deal with this, thinking of the glutamine tRNA, where I showed you that the 4th base pair, identity of the 4 base pair, is critical for promoting efficient subcodon read-through. So it's an analogy. That's why they told me, come to my talk. This will be interesting. So it's, again, the anticodon stem. And again, you need to recode stop codon to tryptophan. So we did simple thing, of course, it's just to show that there is a mismatch. So we restored the base pairing of this Varshney non-stop tRNA. And then we went to the model systems, Trypanosoma brucei and Saccharomyces cerevisiae, where we have 5 base pair as everywhere else. And we mutated the top base pair to unpin it. So we have now wild type tRNAs from Blastocutidia and also the mutant ones. And we went to Saccharomyces cerevisiae or Trypanosoma brucei, the luciferase system, checking what these mutations would do. And I'm just showing one model, so you can actually appreciate that as long as you have 5 base pair tRNA, the read-through is less efficient, much less efficient than when you have this, this unpinned base pair. So this is the Blastocutidia, cerevisiae, where we mutated it. This is the Blastocutidia. This is the wild-type tRNA. When we restore the base pairing, we very much reduce its activity. And this is T. brucei. So this is the wild type. When we unpin it, it goes up. And this was beautifully reproducible also in yeast, in budding yeast. So basically, that's the thing. It does the job. Interesting. But now, still, if you look into the cytoplasm, you would have mRNAs with in-frame UGA stop codons that are with higher frequency recognized by these 4 base pair long tryptophan tRNAs. But there still would be the release factor that would compete with these tRNAs. And the fault was, let's say, 5-fold or 7-fold in some cases. So it's still not enough maybe to outcompete all release factors. So there must be maybe some additional factor that would allow the tryptophan tRNA to reassign the UGA. So we look into the sequence, we compare the sequence of the release factor from Blastocutidia and all of the eukaryotes. And there were some differences, but what was striking was the serine 74G, which is very close to the Nix motif, which is required for recognition of the stop codons. And so it was mutated. And there were one more hint that I don't have time to talk about. So we said maybe it's this serine that's somehow required to recognize UGA, and when it's mutated to G, G is actually the residue in Blastocutidia nonstop. Serine is in all of us. So when the organism mutated it, it can no longer recognize UGA. So we went back to yeast. We deleted the release factor in yeast, and substituted. It's actually 67 position. This is serine to glycine. And we checked it. The cells didn't like it. You can see they grow really, really poorly. So there was the first indication that this residue is important. But when we did this experiments, so we noticed, and this was actually published before, which we realized along the way by Olivier Nami, that this substitution, serine to G, is really restricting UGA recognition. So if you look at the wild type cells, so this is the basal read-through of UGA, UAA, and UAG. So look at the scale, it's very minimal. UGA is known to be the most promiscuous, but it's still, you know, only twofold higher. But if you look here with the mutation, so these guys, they don't change, and this goes up by 100-fold. So this mutation that occurs naturally in this organism is completely restricting UGA decoding. So the next obvious question was, if we combine these, if we combine these two elements, you know, the mutation in the release factor and this 4 base pair stem, would we see an additive effect? So this is the 100-fold increase, as you can see. And when we have the mutation and we add the tRNA, we go to 220-fold increase. With a 5 base pair, we go much lower. So this is enormous increase in UGA recoding by the different tRNA that we believe is sufficient for these organisms to really, because it occurs naturally in this organism, that allows them to recode the UGA as a stop codon. So to summarize this part, I told you that UAG and UAA are recoded by 2 tRNAs that are perfectly complementary to these 2 subcodons, and this actually UGA required 2 things. It required the 4 base pair tRNA plus the mutation in the release vector to make it, you know, to make it go signal for the ribosome. So, and this is not true only for Blastochiaron stop. It is also true for the Condylostoma magnum, which is completely unrelated organism, and it also has all 3 subcodons reassigned in a very similar way. So we could also show that it's conserved, you know, in evolutionarily very distant organisms. So this is actually work of Zuzka and Ambar from Julius' lab. Okay, so this is the breakthrough. Finally, we got it published in a nice journal, and we even made it to the top to the cover. It was quite nice and gratifying. But after this, you know, so we were thinking, okay, cool. So we have these players. So we have release factor with a mutation. We have mutations in the 5th base pair, 4th base pair of the anticodon stem. So there must be something, you know, interesting going on in there. And so if you think of a balance between initiation, termination, and read-through, so we kind of thought that maybe, you know, alterations of the amino acids that are required for recognition of the stop codons in release factors and the basis on the anticodon stem of these tRNAs, they contribute to this balance in a way that really influences the fidelity of the termination and propensity for stop codon read-through. So can we show this? And of course, can we find out why is it so? What's the molecular mechanism? So this brought us back to the original story that was put on hold for nearly 3 years. And so we hit the road at the full speed to answer this question. So this is again Zuzka. And so we hypothesized that there could be, you know, so that the length of the anticodon stem and also the nature of its base pairs could play some role. And if you think of the A side, you know, it's basically a compartment just constricted. So the tRNA comes, and most people focus on the anticodon base pairing, maybe the anticodon loop, but the rest is kind of not overlooked, but it's not the main focus. So we thought maybe the stem of these tRNAs is making some contacts with either ribosomal RNAs or ribosomal proteins, and these contacts could be important for this. So yes, this is actually basically what I say here. So could we find some contacts between the ribosomal, between the basis of the anticodon stem and ribosomal proteins that are, you know, instrumental for this phenomenon that we have seen? So thanks to yeast, we can do a lot of genetics. So basically, we looked into the A side. So this is how the A side looks like. This is the A side tRNA. This is the 4th base pair. And you can see extensions of 5 ribosomal proteins, ribosomal protein 15, 25, 31, and so on and so forth. And so we said, okay, so let's delete them one by one, mutate these extensions, and see if we can get some genetic interaction with our mutant tRNAs. So this is actually work of Petra Miletinova. And so, we started, so this is the approach, the mutagenic analysis. So we started with RPS 30 because there was a hint in the unpublished data from Ron Beckman, who is our collaborator, that this arginine 10 and histidine 5 of the N-terminus of RPS 30 might be important, or might be close enough to the anticodon stem, so they might be important for this tRNA to be efficient in read-through. So we created lots of mutations, always, yes. And so these are so this is again the wild type cells. There's nothing mutated. Then we have cells where this RPS 30 was deleted, complemented by the wild type, so it behaves the same as the normal wild type. And then we have different mutations of the N-terminus. Some of them, as you can see, they improve fidelity. Some of them relax fidelity. So it's definitely important. This protein is definitely important for accommodation of tRNAs. And this arginine, which was supposed to be super important, they didn't show any phenotype. We didn't get discouraged. And then we went on and combined this particular mutation, also all of the others. But we combined this particular substitution, arginine 10 alanine, with the tRNAs. So here again, you see wild type. This orange is the mutant protein. And then here it is combined with the overexpressed wild type glutamine tRNA. And you can see that in the wild type, we have this usual increase in read-through, but with the mutation, it drops down significantly. And when we combine this, when we combine this mutation with this mutant tRNA, we see epistasis basically. So there's no increase, there's no interaction. And as a control, important control, we took another tRNA, which was shown to be highly efficient in read-through, the tyrosine tRNA, which recognizes the same stop codon, and we could not see this. So it was very specific. So even with the mutated arginine, tyrosine tRNA didn't care. So we were quite happy to see this. And again, we could show that this is conserved. And so the obvious question which also came from the reviews was, so, it's arginine. So do you need positive charge? So we mutated the arginine. We replaced the charge, for the negative charge. And if you compare these two things, so this is reduced significantly, but by replacing the charge, we completely killed the activity. So there's no activity at all, even though the protein, everything else works fine, but just this tRNA cannot be incorporated. So we think that it's actually a negative charge, the positive charge of the arginine that can contact the base pair. We are trying to get a structure of this for several years with Roland Beckman, and this charge interaction would be critical for allowing this tRNA to be efficient in read-through. So then we went on. Okay, not on. And we checked also other tRNAs. So I'm not showing the data, but we could show that not only glutamine tRNA, but also tryptophan and cysteine tRNA require arginine and histidine, as was predicted for their propensity for stop codon read-through. Not tyrosine. So tyrosine was puzzling, but with mutations of, yes, so puzzling. So we mutated all of these proteins. And so we found that in case of ribosomal protein 25, when we actually deleted the first, I think, 11 amino acids from its L-terminus. So this is the wild type situation. So we have here wild type glutamine tRNA and wild type tyrosine tRNA. So this is what I showed you before. So they both increase read-through. But when we have this mutation in RPS 25, so the glutamine tRNA is fine, doesn't care, but the tyrosine tRNA read-through goes down. And so it's basically the opposite of what we see with the glutamine tRNA and tyrosine tRNA in case of the RPS 30. So in case of RPS 30, the glutamine tRNA dropped down, tyrosine didn't care. Here with RPS 25, glutamine tRNA doesn't care, tyrosine goes down. So in other words, so tyrosine tRNA also requires some of one of these contacts, and this contact is promoted by ribosomal protein 25. It's N-terminus, not 30. So to put everything together, so we got it published finally this year. And we proposed a model, that should explain why not all tRNAs that are near cognate to stop codons promote read-through, because those that do not, they don't establish any extra contacts within the A site between the ribosomal proteins and the anticodon stem. Only for these 4, they do contact ribosomal proteins, and these contacts actually you can kind of imagine as a holding hands of the ribosome that would, grab onto the stem and compensate for this improper geometry. I told you at the beginning the geometry is important of this mRNA RNA mini helix that would have a tendency to evict the tRNA, not to select it, but because you have this extra force, extra kind of interactions, they allow these tRNAs to overcome this barrier and be selected. So in terms of the kinetics, you can think of the codon simpleton kinetics. So this is when you have non-coding tRNA, it's immediately rejected. When you have a cognate, so it takes some time, but then it's accepted. So near cognates, they are somewhere close to non-cognates, but with these RTI tRNAs, they're getting closer to cognate tRNAs, so they can be also selected. And so this is what we think is the mechanism of their success in doing, in promoting read-through. Now, just a quick summary. So what I showed you is that the wholesale stop codon reassignment in non-stop eukaryotes is possible, and we hope that we explained why. That the alterations of the specific residues in release factors and bases of near-cognate tRNAs actually greatly affect the balance between the fidelity of termination and propensity for stop codon read-through. That we think that we know why only some near-cognate tRNAs are efficient in promoting stop codon read-through, and also that some ribosomal proteins in particular seems to be super important for this because they contact these tRNAs. So this is the basic thing. And now, so what is it, good for, in principle? So there are human monogenic nonsense diseases that, like Hurler syndrome, cystic fibrosis, Duchenne muscular dystrophy, and epidermolysis bullosa. And all these, you see, that are really terrible diseases. It's a pain to look at these people. And so they are all caused by just one stupid mistake in the genome. So it's just one substitution that changes, the sense codon to a PTC, nothing else. Just, it's as simple as that. And so obviously, and actually there's like 11% of all inherited genetic diseases that are caused by these premature termination codons. And so since it's so simple, with respect to the molecular background, it should be possible with a simple solution to fix these errors, and it was shown actually it's possible, and cure these people. And so together with, come on, it doesn't work. So our idea now is actually to exploit all this knowledge and of course the knowledge of all the others. And it's also not only our idea, but now the field is moving towards trying to use all this information, to use these tRNAs as therapeutics that could actually help these people. And so we have put together a proposal for ERC synergy, which is called 3 Stops to Go, with Julius and Olivier and also Mark from the US, who is actually a human geneticist. And I'm showing this just because I'm still excited. We were invited, so we went through the first 2 rounds, and we were invited to the 3rd round. So 2 weeks ago, we were in Brussels for the interview. So in 2 weeks, we should learn if he got it or if he didn't get it, but we had a good feeling. You can see we celebrated already, at least a good feeling in the pub. So please cross your fingers for us, because this will be a big thing for all of us. And that, I think, is all. So this work was started by Petra, and I would not be here talking about tRNAs without her. So her contribution was enormous. Then Zuzka, who took over, then Petra, another Petra. Most of Petras are in my country. And then also Adriana and Clara, who helped. And now actually this project is carried on by Naval Alkami from Lebanon and Filip Brazdovic from Slovakia. So yes, that's it. Thank you.
idea as to why it is repurposed in certain organisms?
Why it is repurposed? That's $1 million question, you know. For example, in this case, this is a typical question that, you know, we ask. So also, how could it happen? You know, because you need, so this organism needed so many changes to occur at the same time to be able to survive, because all of these changes, you know, would be detrimental. And why is it so? You know, for example, in this case, we thought that it could provide this organism kind of a defense against viruses, for example, because you know, this different genome the viruses could not replicate in principle. But the short answer is we don't know. Yeah, there are of course some cases where, you know, we have limitations in the tRNA pool and stuff like this. That this occurred as a need, but for this particular organism, we don't know.
Hey, Elias, wonderful talk. Kind of related to what was being asked there, I'm curious, why do these crazy organisms have so many stop codons?
Why?
Yeah.
Again, the same thing, you know. I think just to make my living. It's all I can say. We really don't know, you know. So we have a theory that that we had to put together also, you know, so that funny thing is that there's more. So we now have at least 4 isolates of this organism from different bugs, different parts of the world, you know, and it's the same. The genome is the same. So either they have a common ancestor that then they spread to the world. There's nothing like that, no intermediate, you know. So it's either normal or like that, you know, because we are hoping that we will be able to find some intermediate which could explain the question, why is it so, you know? So something happened, then some adaptation had to come, another adaptation. So you end up with this crazy organism, but there are no intermediate. They're alike in many ways, and they are from different parts of the world. So I don't think that they evolved, you know, same way in a different parts of the world. Most likely they have a common ancestor, but this missing link is leaving us kind of clueless. Why? You know,
I was just wondering if...
And one more thing, and so with the viruses, actually, it's preliminary, but I think our colleagues that are doing genomics, they could find some traces of viral DNA also. So it's still open question if this is something that now Julius is actually testing in collaboration with, I forgot his name, he's from Austria. So he's testing if these organisms are susceptible to, you know, they have a collection of viruses. So they're throwing all of them at this organism, trying to see if it will be susceptible to infection or not. I don't know the outcome of this, because that could be the one, you know, but it would not be the reason, it would be the consequence most likely. So why? I don't know.
Just a point, I thought if this is telling us something about the evolution of the stop codon, if some of these codons were not stop codons to begin with.
Pardon me,
Sorry, I'm saying if this is telling us about the evolution of the stop codon, that UA, UAG at some point of time were never stop codons and then evolved to become stop codons. Could that be a possibility?
There's also this possibility. We've also explored this, but and I think there's even like a manuscript which is being finished, not by me, but from these guys, genomic guys. But still, there are not so many like real clues to answer even this, you know. So I don't know. Thank you.